Welcome back to the last session of the day, and we will restart with Faelan Mourmourakis, who will present a presentation called Visual Learning and Brain Soup in Tetragonula Carbonaria. The floor is yours.
Thank you. Thank you. Yes, I'm Faelan. I'm from Sydney, Australia. So it's probably really late for a lot of you, but it is 9 a.m. in the morning. So I've just gotten up and I'll be chatting a bit about my PhD work with stingless bees or the Tetragonula carbonaria. Yeah, so first I just want to acknowledge the people, traditional Aboriginal people's land that I've conducted my research on, which is the Wadawurrung clan of the Darra Nation, and just yeah, offer my my thanks that they've let me conduct this research on their land. Okay, so my work is all on cognition in bees. So most people here would probably know bees are very smart. They can do all these really cool things like conceptual learning. They're really good at navigation. You'll hear a lot about how they use the sun to navigate. Really, really big with associative learning. We have so many studies on them being able to do things like conceptual learning. You'll see all articles about how they understand how to count. Really, really cool things. Majority of these studies, though, are done in honeybees or bumblebees. There really hasn't been a lot of work in Australian natives. So I wanted to change that. I had two main questions going into this research, which was how can we compare cognition between so many different species, even just with species of bees. Native bees come in so many different shapes and sizes. Some are solitary, some are social. And also, how do these differences, do they correlate with any cognitive differences, differences in the brain? So today, this is sort of the layout of my PhD. I won't be talking about all of this. I'm just going to be talking about two chapters of my PhD, which is the visual learning stuff I've done with stingless bees and something called isotropic fractinator, which I will get into later. So I just want to introduce you to my stingless bees. This is a comparison photo that I think really shows just how tiny these stingless bees are compared to a honeybee on the left. So they're a very, very tiny bee. They are native to the east coast of Australia. So in Sydney, these are the only species of stingless bee that we get. In Queensland, you get much more different species. Yeah, and we keep them in hive boxes like you would a honeybee. Very low maintenance. They're a very popular animal to keep in people's backyards here in Australia. So that kind of makes them a perfect native bee species going forward to compare to a honeybee because they are also social. They are also in hives. Yeah, so I'll chat a bit about my visual learning with them. So I just did a very simple discrimination learning task. So this is just seeing if they're able to tell between two different stimuli. So I had three different conditions I tested them on. I just did a blue versus a yellow, a horizontal pattern versus a vertical, and a concentric pattern versus a radial pattern. And all these stimuli are very, very popular when it comes to honeybee cognition and bumblebee cognition stuff. So this has all been done to death in them. First time that it's been applied to a stingless bee. So I've taken these stimuli and I've made my little fake artificial flowers, which are just overturned specimen jars and a lot of hot glue. And I've just added, so the little stimuli had a hole, and then underneath would have the reward, which would be 50% sugar solution, or water, which is the neutral stimuli. And this just made, so these are the photos of what that looks like. Very, very dirty. Should have cleaned them before I took photos. But you can see at the bottom, you can see my little tiny little stingless bee ducking her head through that hole and drinking the sugar water or the water underneath. So this just forces them to actually land on the flower and have to crawl to the center and duck in. This is just to make sure that visually they can't fly above and tell the difference between the water or the sugar water. They actually have to put their head through a hole to taste that. So they're not cheating because bees love to cheat. Okay, so this is what it sort of looked like. We made this testing chamber, and the inside we had this white and red pattern. So bees pinks and reds look grey to them. So that's why we've used a red and a white background. And this is just so they don't bump into the walls of a testing chamber. It just creates a lot of visual stimuli for them. And it makes them be able to fly around the area better. So we train them to fly into this box through this hole, and then wanted to just see if they would land on one of the flowers. And they were trained to drink from these flowers beforehand as well. So it took about a couple hours of training these bees before we went ahead. So hopefully we would want them to land on the stimuli that had the reward rather than the water. We wanted to see how long that would take. So here's a little video of that. There she goes. So you can see the bee, how tiny she is, and she's checking out the yellow. So the yellow has water and the blue has the sugar solution. So she's pretty well trained. She's already understood yellow's got the water. She's looking around for the blue. And take her a while to find it, and she's found it. And there she goes. She lands straight away. So they learn very quickly. So these are some results. This is for the yellow versus the blue. You can see that they, so we did about 10 training trials, and then at the end we did a choice test, so an 11th trial, and the choice test was unrewarded. And it was just to be certain that they weren't cheating, finding another way to tell the difference between the sugar water and the water. They were actually using the visual stimuli only. And we can see that they learn very quickly. In just 10 trials, they're getting above 75%. And in our choice test, again, they were getting above a 75. I think the blue was an 80%. So the blue was a little bit better. They learned a little bit better on. But yeah, it was pretty obvious that they were learning, which is great. Honeybees take about 6 to 10 trials also to learn this. So it's very similar to a honeybee. And then this is our vertical versus horizontal. Again, they learned very quickly. In the choice test, they got about a 75%. So they were definitely not picking randomly. And then lastly, we have the concentric versus the radial, rather. And this one was a bit more all over the place at the start. So they were a bit more confused by this one. But towards the end, they were nailing this pretty well also. So this just suggests that they learn pretty quickly. They can do visual learning in the same rate that a honeybee can do, which is really exciting. The next step would be to, which I'm currently doing, is to give them a more complex cognitive task, like a reversal learning that I'm currently doing, and just see how they compare with something like a honeybee and a bumblebee with that. So the next step that I did is something called a brain soup method, which I prefer. The more scientific word name for it is the isotropic fractinator method, but I prefer brain soup. And this method is a way of counting total brain cells in the brain of an animal. And this has been applied to North American Hymenoptera species, but no Australian species. So I wanted to apply it to the Australian species and see how brain cells compared to the honeybee. This method is really, really good compared to traditional methods of getting brain cell counts. The counts are really, really consistent with less than a 10% difference between going from B to B. Very little nuclei get damaged in this method. Probably my favorite thing about the method is that it is so quick. Once I have the brain dissected, so the brain has to sit and fix for about a week. So that's the long part. But once that's done, it takes about an hour for me to go from brain to brain soup to actually having the counts. And the estimates are also independent of brain volume. So I start with a fixed volume. I usually start with 200 micrometers and go from there. So the math is really, really easy to do as well, which I love. Okay, so I'll walk you through what the method actually looks like. So like I said, the first method, I actually have to dissect the brain. That's sort of what that looks like. So the brain comes out. Then we have to fix it in PFA. This usually takes about a couple days to a week, depending on the size of the brain. So for the stingless bees, it's only going to take a couple days. For something bigger, like a carpenter bee or a honeybee, it can take a week. And this fixation just means that the cell walls are more stable. So when I'm actually grinding it the brain down into a soup, it's not destroying those nuclei, those brain cells. And then the next step, once that's been fixed, is to pop it into this tissue grinder and homogenize it. So we do that with sodium citrate and a detergent called Trident, just so we're not damaging very delicate glassware. And crush that down into a brain soup, and that usually takes about 10 minutes to do. It looks very gross, doesn't look like a delicious soup. And next we add a dye to that. So a dye called DAPI. The DAPI is blue, and it will dye the nuclei a fluorescent blue, so I can actually count them under a microscope. And then I load that onto my slide, and then I pop it into the microscope, and it actually glows bright blue at me, and I can count them. So it's the exciting stuff. This is what bee brain cells look like. So on the left, I have my tetragonula, my stingless bees. And you see these little dots, that's what I'm counting. Those are the nuclei of the brain cell. And I think the most exciting thing is on my right slides, you'll see these are the brain cells of a blue banded bee. So I just got these results literally yesterday. And they're extremely exciting. You can just tell visually how many more brain cells a blue banded bee has compared to the stingless bee. And yeah, it looks like a galaxy of stars. It's just beautiful. Very, very cool to count. So I will sit there and I will shine white light as well, which makes my little grid on my cell pop up, and I count all the cells on the grid. Okay, let's do some results. So see my red, my pinky red over here? These are for the honeybees. So I also did some honeybees to start off with because their brains are much bigger, easier to dissect than the stingless bees to start off with. And we also wanted to make sure that the method was accurate, that I was doing it correctly. We know honeybees have just under 1 million brain cells. So we measured brain. So this bottom shouldn't be brain mass. Apologies. It should be body mass. So that was a typo. So the x-axis is body mass. So bees are about 100 milligrams, and they have just under 1 million brain cells. So that's all great. And then our really exciting results were our stingless bees. So stingless bees have about just under 700,000 brain cells. And when you compare body mass from a stingless bee, which is 5 milligrams, to a honeybee, which is about 100, you can just see the difference between brain cells from 700,000 to just under 1 million is not that different, which is really exciting because stingless bee is actually absolutely tiny. Other exciting results are I've only got 2 data points so far for our blue banded bees up the top and our green metallic carpenter bee down the bottom here. So hopefully I'll catch more and I'll get more results there. But you can see our first blue banded bee has well above 2 million brain cells. So that was a crazy big amount for a bee. And our carpenter bee is just also above 1 million. So this is what it would look like compared to North American bee species. So this is a paper by Godfrey that was published a couple of years ago that did this same method with a bunch of North American bee species. And you can see all the colored filled in dots are my results. So Godfrey did a whole bunch of Hymenoptera. So we have our ants down the bottom. Sorry, ants aren't they don't have a lot of brain cells. Our bees and our wasps are doing a bit better. So we have our tarantula hawk wasp all the way up the top. Next we have our carpenter bees and our bumblebees, and then also our wasp in this light blue triangle, and then also our sweat bees over here in the purple. And yeah, you can see, I think the most exciting result is that the stingless bees are quite high for number of brain cells to body mass across these North American Hymenoptera species as well. So the results are so far looking pretty exciting for our Australian bee species. They look really, really high compared to the North American. So we'll see if that's a trend that keeps going. Yeah, and a theory so far is that the solitary bees are going to have more brain cells, which seems to be holding true so far. So the plan is to get more solitary native bee species as well as more social stingless bee species from Queensland and see if they also are around the same as the tetraconula. So that's it for me. If anyone has any questions or just wants to talk about bees in general, I'd love to hear them.
Thanks, Phelan. So we have a first question, I mean, actually two questions that came from AB George. The first one is more, I think, for fun. He asked, like, who came up with the name brain soup?
So it was the professor who came up with the method, Susanna Herculano Honzel. So she has a book. I recommend reading her book. I think it's called The Human Advantage. She talks about how they came up with the method and how they sort of went, okay, we'll call it isotropic fractinator, but we could have come up with sort of a more common, more fun name for it. And they just started to call it brain soup because you literally are taking the brain and you're making it into like a weird, gross soup. And Susanna actually came to our lab all the way from America to Australia to actually teach us how to do the method personally. So that was really, really exciting to actually learn from the professor who created the method.
Mhm. And his second question is, would you not need to pull multiple individual samples if the brains are quite small?
Sorry, could you repeat?
Would you not need to pull multiple individual samples if the brains are quite small?
Yeah. The brains are really tiny. We are doing multiples. That's what the question is. So all of these dots are the multiple different brains. Um, I think that's what the question was.
No, I
Apologies.
think what he's asking is because like very often for this type of analysis, like let's say that if you wanted to do an HPLC or something like this with such a small brain, you will need to pull two or three brain to actually make one sample.
Okay, yeah. With this method, no, it's just one brain, one sample. Um, yeah, I'm quite good at getting the brains out. It took a lot of practice, but yeah, as long as I have the full brain, if any parts of it break, if I accidentally cut some parts off, I can't use it. It has to be a full brain. But yeah, this method uses just the full brain. It doesn't need multiple brains to make up just the one sample. With just how the method works.
Okay. He replied that it's very cool. Thanks for the talk and the answer. I was wondering, like, so the bees that you are showing at the exact at this moment, it's the bees also that you tested for learning in memory, or they are
Uh,
independent?
yeah, so they're at the moment they're independent. I haven't done any studies with the solitary species, so the blue-banded bees or the
Mm-hmm.
carpenter bees. Um, I do. There is someone in Melbourne, Scarlett Howard, who is doing very similar cognitive work with solitary bee species.
Yeah.
The way my method is set up wouldn't work with a solitary species. I need the bee to keep coming back for multiple trials. If a
Mm-hmm.
solitary species, they're just not going to do that for me. I think going forward, I would like to work with more people to get cognitive research for the solitary species. But just at the moment, I have a year left of my PhD, so I don't have the time to do it, unfortunately.
Okay, I see. And like to go back to your protocol, I was wondering like if the stingless bees also deposit some chemical when they are feeding on the flower,
So they
like
do.
honeybees or bumblebees.
Yep. So they do. They're very, very big into like that. That's actually how I train them compared to the honeybees. Is they really, really big on the pheromones, on the footprints that they leave on the flowers. Between every trial, we are replacing the flowers. We're washing with ethanol
Hmm.
just to get rid of that completely, because otherwise, they would just start to use that pheromone rather
Yeah, no,
than
of course.
the actual visual. So yeah, they are
Yeah.
being cleaned.
Okay, okay. And I was wondering if you observed any interesting color preference, like that could differ a little bit from what we are usually observing in honeybees or
Um, Sorry, the question was, are there any differences between the stingless bees and the honeybees in terms
no,
of?
like if I mean like yeah, because we know for instance that honeybees will more go for blue, but like did
Yeah,
you observe this type of preference also in stingless bees or not?
yeah, they prefer blue a tiny bit more, not a significant amount. That's kind of because I have done similar work for my master's with honeybees, so I've done very similar experiments with them. I would say it's pretty similar to the honeybees' preference in blue, is they also have a slight preference for blue, but it's really easy to train them out of. Like they once they realize yellow is going to be the reward, they get over it very quickly. But just if go back to the results, a lot of transitions. Sorry. Yeah, so you can see here that they did have a slight, especially in the choice test, they had like a 5% more preference towards blue. Like even at the start in the first trial, majority of them would just pick blue straight away. So they did have that slight preference.
Did you already think of like making your protocol according to the first choice? Like meaning that if the bee is choosing the blue, you are rewarding for the yellow.
Yeah.
And opposite.
Yeah.
Like this, you would avoid this strong effect at the beginning, which can give the feeling that they are not really learning.
Yeah, definitely. Look, my worry with doing that was that I would have too many of one condition compared to the other. I wanted it to be pretty equal. I wanted, I did 20 trials, 10 trials on the blue being the preference versus yellow being the preference. I feel like if I had done that, majority of it would have been, one or the other, just because they did have that slight preference. Another issue was also time. We had a really, really bad season in Australia last summer. We had floods, we had everything, we had rain, so I had a very limited amount of time to get a good amount of data. So I had to make the choice we're going to try and keep it as equal as possible and do half blue, half yellow. But going forward, if I have time this season, that would be something I would like to actually look into a bit more and try and see
it's
if
a
that
method
protocol.
it's a method that I myself used, and it's kind of easier because like you're winning a bit of time. Okay, so like we are done for your talk. Thanks a lot.
Wonderful. Thank you so much.
And like everyone, if you have more questions, like do not hesitate to go on the dedicated channel of Faelan on Discord.
Wonderful. Thank you so much.
Thank you.
Bye-bye.